Check it out now, y'all.
nanoHUB-U online instruction.
Hello and welcome back to another lecture of nanoHUB-U's course Introduction to Bioelectricity. In this second week, we are talking about the chemical basis of electrical signals, and in this third lecture, we are going to be discussing ion channels. So in the last lecture, we talked about active versus passive conduction, saltatory conduction, myelination, and so on, and all of this relies on the presence of these ion channels. Ion channels are also important at the interface between neurons, where one neuron wishes to communicate with the next neuron. So the signal, the action potential traveling down the neuron reaches the tip of the axon, and something happens, which we'll discuss in subsequent lectures. Something happens which releases neurotransmitter into the synapse, and that causes ion channels, different sorts of ion channels, to open or close in the postsynaptic neuron. And so in both cases, you've got these ion channels. And you see ion channels actually in a number of different locations and applications throughout the human body. So we're going to begin to talk about ion channels, the sorts of ion channels that you see, the ways in which we look at ion channels and we explore how they work, and what we learn through this whole process. We'll do that today. So let's jump right in and talk about patch clamping. All right, so I talked about patch clamping briefly in the first week. And patch clamping is this technique that was developed in the 50s and 60s, whereby you take a glass capillary tube and you hold it over a flame and you pull the tube as it begins to melt, and that causes the glass to stretch and narrow. And if you pull it just right, and then you cool it and you break it off, you can get a tube that has an inner diameter of just a few microns. If you polish it off and then you hook it up to a stereotactic rig or an electrophysiology rig, you can then approach it to a neuron that you're studying on a dish, for example, or maybe a slice of brain that you're studying in a dish. And if you are delicate, you contact the surface of that cell without breaking the membrane. And when you do that, you apply a small amount of suction, which creates a seal, a high impedance seal, around the interface between the pipette and the cell membrane, and that creates an insulated electrical environment that allows you to record the activity through an individual ion channel or a small number of ion channels. So you can imagine that in a typical neuron, you could do something like this. You could seal around a particular patch of membrane. You might find that you have just one ion channel, and then you can study that. You might find that you have a large number of sodium and potassium ion channels, in which case you could potentially use a drug like tetraethylammonium to block off the potassium ion channels, and then you could study the sodium ion channels in isolation. Or you could use tetrodotoxin to do the opposite. So you can begin to play these games and look at ionic conductances in the neuronal membrane. But there's more. So there are a number of different configurations in which you can use a patch clamp. After you've patched onto a cell, you can apply more suction until you tear the membrane without losing that seal. And that allows you to measure the electrical activity inside the cell without interference from the outside of the cell, without the cell leaching or leaking out. So you can measure the electrical activity inside the cell. If the cell is electrically small, that allows you to do a whole cell recording. If the cell is electrically large, like a very long neuron, it allows you to do a local recording of the intracellular environment. You can also, before rupturing the membrane, but after creating the seal, you can physically pull back on the pipette and tear off a section of membrane without losing the seal, and that allows you to do an inside-out recording. So you can apply different sorts of chemicals to the inside of the membrane. Some of these ion channels we'll learn are activated from the outside, some are activated from the inside, some are both. But you can apply different chemicals to the inside or different substances to the inside of the cell and see what effect that has on the intracellular current through this ion channel. You can do an outside-out recording, where you first tear open a hole to do the whole cell recording, and then you pull away, and these two segments of membrane anneal together. And if that happens, then you're now looking at the inside of the membrane, and you can modify what you see on the outside. So you can look at intracellular currents for various different substances applied to the outside. So it's a very, very powerful technique that really allows you to explore what is going on in the neuron, and it's key to a lot of the things that we're going to talk about today. A lot of the discoveries we're going to talk about today were learned using this technique. So, individual ion channels. All right. If we look at a membrane potential, so if we have a depolarizing membrane potential going from minus 80 millivolts up to some higher voltage, higher than the threshold voltage, so that the sodium ion channels will begin to open. And if we record microscopic currents from various different points on the cell membrane, you'll find that at any given ion channel, you'll have some amplitude of fixed current that you expect to see. So some small current. And you might record from a variety of different locations and see a variety of different currents. And if you were to aggregate all these together, so if you're zooming into specific small areas of a neuronal membrane, if you add all these together, you would expect to see a sum of these various currents. And if you summed all of these transient inward currents, you would be able to reconstruct the sodium current, which looks a whole lot like a small version of the transient inward current we see during an action potential. So all of these individual signals add up to the transient inward current, and that confirms for us what we had expected, which is that it's the sodium ion channels that are leading to the transient inward current, and it's the transient inward current that accounts for the depolarization of the neuron during an action potential. Furthermore, by looking at the relative positioning of all those little currents, you can tell when they are opening as a function of voltage. So we talked earlier about, in 2 lectures ago, we talked earlier about how these individual currents represent particular thresholds, and these thresholds are all set around a specific value, but there's some variation. There's a distribution around that value. And so if you take the distribution of the opening of channels as a function of voltage, you'll have a Gaussian curve. And if you plot that curve with the probability of the sodium ion channel on the y-axis and the membrane potential on the x-axis, you'll see that curve shown in a different format, which is that as you move in voltage from low values to high values, you have a region in here which contains the threshold for almost all of the sodium channels. And then you still have some channels opening up early and some opening up late, but the bulk happens in here. So if you want to depolarize a neuron, you can come right to the threshold voltage, but you'll have a more rapid depolarization if you exceed it, so that you capture a higher density of the sodium ion channels that you want to capture. You can do the same sort of experiment with potassium channels. So with potassium channels, you can stimulate with a fixed pulse. You're going to get an initial delay, which you can see here, because this is, after all, the delayed outward current we're looking for, right? So you're not going to get potassium channels opening initially, because the first thing that happens during action potential is that the potassium channels close. Then the sodium channels open. Then there's a delay, which we'll understand better when we get to Hodgkin-Huxley, and then the potassium channels reopen. And when the potassium channels reopen, you start to get these currents, which you can add up. And if you add them up, you see a microscopic current that has a shape very similar to the delayed outward current that we talked about 2 lectures ago. Here's the delayed outward current. You can see that they match up very nicely. And this represents the same sort of probability distribution for potassium ion channels closing opening, as a function of the membrane voltage. So these channels that are opening and closing, they come in a variety of different forms. And what I mean by forms is not their protein-based structure, though that's very important as well, and that will give you a better understanding of how these channels function exactly. But if you're interested in that, I really recommend you take a neurobiology course. For the purposes of this course, we can understand broadly that there are 2 sorts of channels that we are going to talk about again and again. And then there's a few other sorts which I'll introduce today. The first sort of main category of channels that we'll talk about are voltage-gated channels. That means that when the voltage exceeds a particular threshold, these channels open or close. You can have voltage-gated sodium channels, voltage-gated potassium channels. These are the 2 that are involved in regenerating an action potential in an axon. You also have voltage-gated calcium channels, and voltage-gated chloride channels. And these are involved in a huge variety of different ways throughout this whole process. And we'll get to some examples of that later on in this lecture. The second main category of ion channels that we'll talk about are what are called ligand-gated ion channels. So ligand-gated channels are not opened by a voltage. They're opened by a particular molecule or ion that binds to the ion channel and opens it up. It can bind to the outside of the ion, as happens with a neurotransmitter receptor. It can bind to the inside of the ion channel, as happens with calcium-activated potassium channels. Or it can be a situation in which you have a secondary messenger. So a neurotransmitter that binds somewhere else on the membrane releases a series of cyclic nucleotides, like for example cyclic GMP, that then binds to a number of different ion channels, opening them up. And we'll talk more about those secondary messenger ion channels in the next lecture. But for today's purposes, we have a number of different ligand-gated ion channels as opposed to the voltage-gated ion channels. So let's talk about other types, right? We've talked about voltage-gated for the past few lectures. I've introduced ligand-gated. We'll talk more about them, but there's other kinds as well. So you have these stretch receptors. So when you touch something, right, there are organs that are built into your fingers and everywhere in your skin, but they're most densely populated in your fingers, that allow you to detect the surface roughness of something or the amount of pressure with which you're pressing on something. That happens through what are called these mechanoreceptors, which are buried throughout the skin. And there's many different kinds. The Meissner corpuscles and the Pacinian corpuscles both detect spatial frequencies. So when we talk about auditory frequencies, we talk about, a sine wave and what is the frequency, how fast that wave goes up and down. The spatial frequency is the same thing, except instead of being in time, like an auditory signal, it's in space. So if you think about sandpaper of different roughnesses, a lower number, a rougher sandpaper, has larger particles spaced further apart. That's a lower spatial frequency. A smoother sandpaper, higher number, has smaller particles spaced closer together. That's a higher spatial frequency. They occur more often in space. When you rub your finger on something to detect the roughness of it, whether it's your sweater or the palm of your hand, you're going to be looking at different spatial frequencies. Meissner corpuscles are optimized for low spatial frequencies. Pacinian corpuscles are optimized for higher spatial frequencies. Merkel's discs are optimized for sensing pressure. Ruffini corpuscles are optimized for sensing tension. But these are the least well understood of all of the different mechanoreceptors that you're going to look at. In each case, what you have is ion channels, right? So if you look at a Meissner corpuscle, for example, you're going to see ion channels where the ion channel itself, when you stretch it or when you press on it or you deform it in some way, physically opens. So it's mechanical. It's not voltage gated. It's not a voltage that's opening it. It's not ligand gated. It's not a neurotransmitter binding on it that's opening it. It's mechanically gated. It's physically being opened up. And when it opens up physically, sodium ions rush in, the cell depolarizes, and you get an action potential in the neuron that is adjacent to that sensory organ. So it's a different kind of ion channel. The sense of taste works through ion channels as well, but in this case we have one of those secondary messenger ion channels that I mentioned that we'll talk about more in the next lecture. In this case, you have different senses of taste, right? You have salty, you have sweet, you have bitter, and you have umami, right? These different senses of taste bind to a receptor on the outside of your taste buds. That receptor releases a G protein. The G protein goes through a cascade, which leads to the release of neurotransmitters inside the neuron, which opens up the ion channel, allowing the cell to depolarize and to create an action potential, which the postsynaptic neuron here sees and conveys back to the central nervous system. You also have ion channels which are receptive to temperature. So in temperature receptors, which are closely tied into the nociceptive system, the pain system, these ion channels open at a particular temperature. So if you expose them to a heat of 43 degrees Celsius, these ion channels will open up and they will allow sodium ions to flow in, calcium ions to flow in, and that will depolarize the cell and lead to a cascade of events leading to an action potential and the perception of pain. Interestingly, there's also a molecule called capsaicin, and capsaicin is the primary ingredient in hot sauce and chili peppers. It's what gives food the subjective experience of burning. And capsaicin comes directly through the neuronal membrane, and it binds to the neurotransmitter on the inside, opening it up and allowing the same process of depolarization, which is perceived as temperature. So the reason that when you eat spicy food, it tastes hot rather than cold or salty or anything else, the reason it tastes hot is because the neurotransmitters, or the ion channels that are being activated are the same ones that are activated for heat. They're just being activated in a different way. Rather than activating them by raising the temperature above 43 degrees Celsius, you're activating them by opening them up from the inside of the cell. Another type of ion channel is the hair cell. So inside of your ears, in the cochlea of the ear, you have these hair cells, and these hair cells contain these ion channels, which look a lot like, conceptually, a normal ion channel with a lid on it, where that lid is attached with a protein string, which we draw here as a spring, but it's attached by a protein to the next one of these protruding hairs. And what happens is that when sound resonates at a particular point in the cochlea, these hairs vibrate back and forth, and that bending of the hair opens the trap, allowing ions to mechanically be free to flow in. Diffusion drives them into the cell, depolarizing the cell, leading to these calcium channels, which are voltage gated, opening up, allowing an influx of calcium, which then causes the neurotransmitter to be released. And we'll talk more about that in the next lecture. But for now, here's another type of ion channel that is mechanically gated. Interestingly, you have these in your cochlea, and your cochlea is shaped so that it goes from one diameter to a smaller diameter as you move through the cochlea. The larger diameters respond to lower frequencies. The higher diameters respond to higher frequencies. The smaller diameters respond to higher frequencies. But there's another set of hair cells that are on the outside of the inner set of hair cells. And what these do is they don't pick up sounds. In fact, they generate sounds. So in some cases, your cochlea will create sound, and you can pick this up with a microphone. And some people suspect that that might be linked to tinnitus, this perception of sound when there isn't any. But the real purpose of that second level of hair cells is to actually dynamically stiffen the membrane of the cochlea and allow you to change the gain, the resonance, the ease of resonance of a particular frequency at that point in the cochlea. So you have what we in electrical engineering call automatic gain control built into the way the cochlea processes sound. And that's all a function of these specialized cells, which occur in other locations in the body as well. Like, for example, the vestibular canals, where we talked about the vestibular ocular reflex in the second lecture of week 1. A final type of ion channel that I'll talk about in this lecture are those involved with sight. So you have rods and cones inside your eyes, and those rods and cones respond to light, and they respond to light and to darkness by a cascade that involves these ion channels. So you have this effect where darkness leads to elevated levels of cyclic GMP. That's that cyclic nucleotide we talked about earlier, which causes ion channels to open and sodium to flow in, depolarizing the cell and leading to action potentials being fired through the bipolar ganglion cells and then out of the optic nerve and through the ganglion cells and to the primary visual cortex and the occipital lobe. If you expose the same rod to light, and cones work in a very similar way, if you expose the same cell to light, what happens is that the light processes the cyclic GMP, so breaks it down so that you have a lower level of cyclic GMP inside the cell, which leads to the ion channels, the sodium ion channels closing, which then allows the potassium ion channels to restore the resting membrane potential, and the cell hyperpolarizes and stops firing. So interestingly, we always talk about seeing light and not seeing in the dark, when in fact we do exactly the opposite. We see dark we don't see in the light. So what I mean by that is that these cells fire continuously in darkness. They stop firing when they're exposed to light. So unlike hair cells, which fire in response to sound, taste buds, which fire in response to the presence of a particular taste, the heat receptors, which fire in response to capsaicin or in response to an elevated temperature, rods and cones inside of the eye fire in the absence of a stimulus and stop firing in response to light. So it's backwards from what we're used to. And that's a quick overview of different kinds of ion channels and where we find them and how they tie in to the senses. But one of the nice things about the textbook that you have for this course is that in addition to covering the material that we're covering in lectures, certainly for the first 2 weeks of these lectures, it also has more in-depth chapters about each of these different aspects of sensation that I mentioned today. So if you're interested, I encourage you to go and read chapters on sight or on hearing or on pain or on motor control to see and learn more in depth about how each of these aspects work. In the next lecture, we'll come back and we'll start talking about postsynaptic receptors. I'll see you then.